I am really pleased to be hosting Dr. Ivette Cornejo Corona to discuss her recent PLOS ONE Lab Protocol paper entitled Method for Isolation of High Molecular Weight Genomic DNA from Botryococcus biomass and the associated detailed protocol in protocols.io. Ivette Cornejo Corona is a postdoctoral associate under the supervision of Professor Timothy DeVarren in the Department of Biochemistry and Biophysics at Texas A&M University. She earned her master's and PhD in plant biotechnology from Cinvestav IPN in Mexico. Her doctoral research focused on elucidating stress response mechanisms in a non-model microalgae. Ivette developed protocols to assess stress in vivo and correlate metabolic accumulation in Botryococcus brownie under stress conditions. Her interest in non-model microalgae led her to Texas A&M University, where she applies biochemical and synthetic biology approaches to investigate the biosynthetic pathways of C40 hydrocarbons accumulated in Botryococcus lycopodienor. Ivette's research is supported by grants from the National Council of Science and Technology in Mexico and the Japan Society for the Promotion of Science. She has recently published part of her findings in peer-reviewed journals and received the Achievement Awards for Excellence in Research from Texas A&M University. Ivette, thank you so much for being here.
Gabriel, thank you so much for inviting us to make this. I know we're taking extra time from your time, but we appreciate your effort and we are very happy to be here with you.
No, it's a pleasure to be talking with you about science. So to kick off the interview, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, sure. So this protocol is essentially, it provides a detailed methodology for extracting high molecular weight genomic DNA from the 3 species of Botryococcus. Specifically, is meant for sequencing purposes. We use it to obtain, enough DNA with enough quantity and quality for downstream the genomic work. That is published in another paper. You can go and check it. Which essentially in this other paper, we needed to do like a Botryococcus genomic comparative study. So we needed to have a very good sequencing results. And for that, we need to have, a very good genomic DNA with very good quality. So this protocol is relevant because of that. We showed that we had enough DNA to be used for high quality DNA sequencing. And, this high molecular DNA isolation is a little bit challenging for species that are not model or that are not like standard in like all of the labs, which is Botryococcus case. There is not a lot of people doing this kind of research because it was a challenge about extracting the DNA. So essentially what we did was to optimize a method that was available for everybody and that give us the resources we needed to make the genomic studies after this. So I wanted to go a little bit more deep into what is Botryococcus. So formerly, we used to call Botryococcus we used to have Botryococcus into the 3 races. So race is not the best way to define different species. However, when they were discovered in the 80s, they were just classified as different races depending on the hydrocarbon they produce. So we, since some years ago, together with some other collaborators, we always talked about, make a genomic comparison and give to the community more information about saying this is not a race, it's not just a single species, they are different species, not just because the metabolic results that we can see by looking into the hydrocarbons, but also because they have like morphology differences. So we needed to do this, and in order to do that, we needed to have genomic high molecular genomic sequence. So Botryococcus essentially they like 3 species now. The 3 species are colony forming. They 3 of them are of course green microalgae. They perform photosynthesis for like whoever is listening and had no idea what is a microalgae. They are microorganisms able to do photosynthesis. So Botryococcus is a colonial form algae, and they are surrounded for something we call extracellular matrix. This is composed from hydrocarbon links together with a lot of polysaccharide that we call fibril sheet, and these hold together the colony. So essentially, a way that I see it is that single algae is surrounded for something similar to a sponge that is full of hydrocarbons, and they are all holded together in the between of this sponge. So that makes challenging the extraction of the DNA, because the DNA is going to be inside of this extracellular matrix that we have to take out or to remove, and it's going to be inside of the cell wall because it's a vegetable cell. So we need to disrupt this in order to open it and expose the DNA. That's why it was challenging, because DNA is going to be soluble into the polysaccharide. So that's why also like a lot of the convenient kits that we can get from the market were not useful for this species. So that's why essentially we need to do something. We need to make like a good protocol, optimize it, and think about the morphology that we know because we are who is working with this species, and also to take information from, like other methodologies and essentially work with the troubleshooting. So yeah, that's why this is relevant. And that's why we decided, like to make a special paper to share the details, to give enough detail, and also to open the community to use it for other species of not just microalgae, but maybe for another plants or another tissues that are polysaccharide rich. So yes, that's why.
So just to recap a little bit and to see that I understood correctly. So to be able to perform genomic studies, you needed high quality, high molecular weight DNA. But the challenge here is that because the cells of Botryococcus are covered in polysaccharides, it is technically challenging to extract this DNA purely and mostly intact.
Yes, and yes, mostly intact with a long, because you require enough quantity, but also that have like a good amount of heavy DNA, high molecular weight. So extract DNA is not that challenging. You can do it, but they're gonna be,
Fragmented.
break it. And you don't want that too long sequencing reading. You want as long as you can. So one of the steps that you need to pass through, let's say it, is kind of a quality step that you have to pass is when you submitted your sample, they're gonna make a task in order to let you know exactly how much high molecular weight of DNA do you have, and how good is in terms of how big are those DNA that you send to the company. In this case, we use a company. So you have to be sure that you are doing a good job and trying to keep it as intact as possible. The only thing that you will be able to do at the lab is going to be just to check, more or less the weight into the agarose shell, for example, and you can get a ladder with a very heavy special, like bands to tell you more or less. So what we were looking to have, it was like a good amount by looking for this thick band in the very top that is over 14 kilobases.
And what would you say were the limitations of the previous protocols that existed to extract DNA from similar cells as Botryococcus?
I would say that they are not similar cells. Well, yes, cells, yes, but not colonies. There is, like not a way to you to separate the colony, I'm sorry, the cell from the colony, other than disrupting the extracellular matrix. So there is not a way to do that, essentially. Previous protocols or another protocols or commercial protocols, I don't think they worried about keeping high molecular weight DNA. They are protocols in which they use similar buffers we use. That like the technique seems to be very standard, but it's not, because it was optimized and adapted into the 3 different species we use. But essentially was that, like there is not another cell like this one. I can say that they are another algae with a similar composition in terms of polysaccharide production. For example, there is like Hematococcus pluvialis, for example, there's another colony algae. They grow like in colonies that form like lungs, like chains, and they are covered for a small amount of polysaccharide. So that small amount is easy to take, without disrupting a lot the cell and without, like breaking the DNA at the end. So we needed it to make a pre-treatment to the cells. And that was my idea, I guess. My idea was like, we cannot go to the extraction point without taking the polysaccharides first. And we need to find a way to do it that is gentle with the cells, but good enough to take as much polysaccharides we can before going to the actual process of breaking the wall, the cell wall, and extracting the DNA. So essentially, that's why I came out with these, let's say, steps that were useful to go and take the polysaccharides before going to the extraction point. Sorry, I forgot about this. My sensor is far away. It doesn't, like sense that I am here. But anyways, yeah, so that's essentially why there is not another cell, essentially. There is not another colony like Botryococcus species. And that's why we need to came out with a special process, and that, like it's useful for this. We did sequence before. They are genomic sequencing results before this new one. They were not with long sequencing, and they were very like short readings. And short readings are not good for have a good, it's not good enough to make genomic comparisons. So that's why we need to have something new and something with with more innovation, let's say it.
And your response naturally leads us to the protocol itself. And you already mentioned some of the most critical steps. But can you again, for the audience, summarize what do you think in your protocol are the most critical steps?
Yeah. So I would say the very first step, if you go to the protocol, they are like this diagram that I try to summarize 6 steps. And the first one is the biomass preparation, which is essentially very standard for any kind of biomass, right? We collect the cells and we freeze dry immediately. That's important. Freeze dry and keep freeze the sample are important because that's gonna give you a better quality of DNA. That should be one. The other one is the pre-wash that I already kind of mentioned a little bit. So this step for the Botryococcus species was completely, let's say, it was perfect for what we needed to do. So what we use is sorbitol, which is a sugar. So this sorbitol is actually going to dissolve the polysaccharide that is outside of the cell. So it's going to try to remove. So this step is the most critical one to me. And for that, you need to be sure that you are making the homogenization very well. One of the other things for people that have no idea how Botryococcus is, like this extracellular matrix and the hydrocarbons are going to make the cells to float. They're going to be floating. So when you make, for example, the centrifugation, you're not going to have the cells in the bottom. You're going to have a very thick layer floating in the top. So something very critical and something that people needed to realize when they are running with this protocol, when they're working with this protocol, is that when you do, so you have to add the sorbitol solution or the pre-wash solution into the cells. You're going to homogenize with a sonicator. The sonicator step is also very critical. I'm not using a lot of energy. I'm using very little, and it's in order to try to break the extracellular matrix, to break the fibrils. So it's very short and very quickly. And because of the results, I know it's not going to interfere with the DNA because I'm not breaking the extracellular, I'm not breaking the cell wall yet. I am trying to dissolve the extracellular matrix. So I have the cell and I'm trying to remove everything that is surrounding the cell wall. So what you have to do is just add the sorbitol solution or the pre-wash solution, make sure that it's very homogenized with the pulse of sonication, and then you incubate it and you make the centrifugation. Something that is important to notice in this step is that every time, so I repeated 3, 4, or 5 times this pre-wash step. Of course, with more times you do this, also you can maybe have a little bit of cell wall disruption. So it's better to realize that after every step is a centrifugation step. So the thick layer that I mentioned a little bit before is going to start making less and less, and the cells are going to be going to the bottom of the tube. That's the indication that you're doing a good removal of the extracellular matrix, and that the polysaccharides and the hydrocarbons are actually being dissolved from critical. Of course, it will depend. For example, if someone wanted to use this protocol for another tissue, they need to realize this difference because it's important. Again, there is not, like other species with this specific characteristics. If you have a tissue that is also with a good amount of polysaccharides, you need to realize if this one is not floating like Botryococcus, maybe 1 or 2 washes or pre-washes are enough. They needed to, like check out the differences. The other critical step, so after that, you have like a clean biomass with a minimum amount of polysaccharides. So then you can break the cell wall, expose the DNA, and then dissolve the DNA, the acid, the nucleic acids. And then after that, we have a step that is an step that is called RNA removal. So we don't want RNA. Of course, this protocol is to get, this part of the protocol is to get all the nucleic acids. So in this other step is the RNA digestion. So essentially what we do is to add RNAs, and then later we clean, we remove the RNAs and the RNase enzyme by using chloroform, which is very, like used technique. And then after that, we incubate and we resuspend. The resuspended step, which is the very last one, is also very important and is critical because usually what we do is to put into the vortex to resuspend to be sure that everything is mixed. In this case, it's better to not to. It's better to, like add the buffer for the resuspension, and by pipetting slowly, like kind of mix it, go to the walls and leave it a little bit at the room temperature, because otherwise you could break the high molecular weight DNA. So essentially is that. So if I go back, it will be the biomass, the very first step. If you don't have a good preserve biomass, your protocol is going to be useless. So that's a very critical one. The pre-wash that you need to optimize if it's necessary for your own tissue, if someone else wanted to do it, the RNA remotion, and the last one is the washing and resuspension, last step.
And as you were developing the protocol, was there anything that you tried that didn't work and didn't make it to the final version of the protocol?
Oh, yeah. Yeah, so Dr. Boland, which is the first author in the other paper, he was a former student here. I will say he tried several different combinations on the extraction buffer. We use CBIT, which is very standard. And you can look and you're going to find like at least 5 or 10 different versions of, ratios between the CBTA and the other components. He tried for, I would say, a couple of months with different combinations. And this one, when I started, what I did was to include my idea of the pre-wash before trying with his buffers. So I used his combination that was the better, but not perfect. And so we combined ideas and we, ended up having this specific recipe for the buffer.
So your protocol is relatively young. Your paper is relatively young. You just published it the summer of this year. But I wonder whether you have made any further optimizations to the protocol since the time of publication.
So we haven't. I did optimize a couple of minor details. So there is not a major detail that I came out with. The protocol as it is working. Actually, one collaborator used it recently, and he used it pretty much identical to this because he's working with Botryococcus biomass. I suggested another collaborator to make a couple of adjustment and the ratio of sorbitol at the beginning, but no, I mean, we haven't done much changes. And something that I wanted to try, I haven't had the time and we haven't had the chance to, set up an experiment that, to do this is I want to try to see if the protocol, it will be good for very old biomass, with old age or young age, because the amount of polysaccharide, it will be different. And the reason why I wanted to do this is because we have the intention of eventually make probably transcriptomics stuff. So we wanted to have, other genomes using different ages cells, but that idea is in the air, and until I have everything set up and with the resources in order to send to the company for them to try and to give us this first result, which is the analysis for the quantification and the quality. I haven't, we just haven't done it yet, but it's an idea, and it could be possible that we needed to optimize a couple of at least a pre-wash, I guess, the pre-wash step.
You opened up the interview by saying that some sequencing was done actually in the paper that you published previously to the protocol itself. And in that paper, you tested the protocol in 3 different now species of Botryococcus. But I wonder whether you think that the protocol can be easily optimized to other green microalgae species. I'm aware that you already mentioned that that Botryococcus is pretty unique in its characteristics. So how do you think this protocol could be optimized to other microalgae?
I mean, it's just, it can be used for sure. The sorbitol, the wash, the pre-wash step, again, it was the one that made the difference for us, and that is giving us this very cool protocol. This one, it may need to require the optimization for different species, and it will depend on the species itself. As I mentioned before, I just thought of Nematococcus pluvialis because it's the one that I work with, but it's Desmodesmus, and they like some other colony forming microalgae that I'm sure they can be the genome. I'm sorry, the high molecular weight for the genome sequencing, it could be done by extracting the DNA using this protocol for sure. And in terms of optimization, again, as I say, we suggested the pre-wash to do it at least 3 times with 3 species of Botryococcus because of the amount of polysaccharides. It's very big. And also the age of the biomass we use is in a, like in the peak point. So it's a lot of polysaccharides in there. But if you use a species like microalgae species with less amount, maybe you don't need to do it 3 times. Maybe it's necessary to do it at least 1 or 2, but that need to be done, like to evaluate. And it's very easy to see, as I say, if you run an agarose yield using this special ladder with the extra bands in the top with high molecular weight, you can more or less say like, oh, well, I'm having better or more high molecular DNA if I use one pre-wash, 2 pre-wash, or 4 pre-wash, for example. Other than that, the extraction buffer is pretty standard. It's not like the ratios we use are not completely like out of the mind. They're very standard. Maybe worth it to check if someone else already did genomic. I'm sorry, what I am saying, high molecular weight, or if they try to extract genomic DNA from that species, just to check what they use to see if maybe they wanted to try. But it's a matter of, spend a couple of days on troubleshooting and optimizing, but it's possible. And I don't see anything that is not something that people can do in the lab if they're working with a molecular biology or stuff.
As we have mentioned a couple of times already, this protocol is actually part of a research paper in which you and your colleagues reclassified Botryococcus brownie chemical races into separate species based on comparative genomic analysis. So my question is, what was the motivation to send the protocol itself to PLOS ONE to be peer-reviewed as a standalone methodological paper?
Well, I'm going to start saying that we choose PLOS ONE because it's an open journal. So because we wanted to share this with as many researchers as possible, not just the comparative genomic, because we are reclassifying Botryococcus, right? Originally, it was just 1 species in 3 races. Now, what we are saying is not 1, it's 3 different independent species based on all the comparative that it was published in the other paper. So we felt that the optimized protocol we have is a value and it stands alone as a resource for other researchers and for other people that is working in this. Again, we are addressing on one common challenge, which is high molecular weight DNA extraction. So this is a good resource for other people. So by publishing it and the PLOS ONE, we kind of aim to make it accessible for another researchers, of course, who may face the similar obstacle that we have before by isolating high quality DNA from microalgae or other species. So this protocol or the protocol's publication is actually stands a lot and is a very reliable reference that can support and provide other people that is working with a genomic and molecular biologist that is like way beyond the specific like application we follow or the specific comparative analysis we have.
And talking about the peer review, how did the feedback that you received from the peer reviewers at PLOS ONE shaped the final version of your protocol?
Oh, well, it was really invaluable, like it's always good to have extra eyes on your research because they may catch stuff that you didn't do. So we had a great time on the check-in. We spent time and we improve our protocol and we make it more clear. The feedback essentially was like, oh, even us more details and stuff. So we worked on that and we ended up having, after the feedback, a greater clarity and more reproducibility, let's say it. And the reviewers suggested additional more details into the protocol. They also go into the first figure on the protocol and they went like, oh, what if you do this or you do that? We are also using the I protocol. Before this, I had no idea that existed. Now I am mentoring because I found the format itself very easy to follow. If you follow the formatting, it will be easy for someone else. So it's kind of a good standardization on protocols because sometimes we are not good enough on giving the details that people need in order to make it more reproducible. So yeah, the input of the reviewers was invaluable, as I say, and I think it make the protocol like more user friendly and more comprehensive.
And again, you published the paper relatively recently, just in the summer. So this question might be speculative at this time, but what do you think has been or will be the significance of publishing this lab protocol paper for your young career?
I'm laughing because you say young. So yeah, I think this is my first protocol publication. Again, I came out with a couple of ideas, and that's why I am the first author in this one, because essentially was most of the work on the protocol was mine. I have few few years of experience working on Botryococcus. I've been working with this species since I was a PhD student. So I know the cells, I know how they behave, I know how they look when I am trying to do extractions or stuff. However, publishing a protocol was something new for me. And I think I feel more strong, more strange on my expertise in the way of communicate myself and communicate my ideas. Sometimes we have a lot of ideas and we cannot share it with other people, or maybe we can, but not in the proper way. So I think that's the first one. The other one is microalgae genomic field is something relatively new. And I think it will contribute into my reputation as a researcher, and not just in the field of microalgae, but in biotechnology and molecular biology in general. Also, it's been allowing me to collaborate with other researchers that they reach me out, that they are, interested or they're trying to apply similar methods, and they have a few, challenges that I already passed through. So this expertise kind of enhanced my skills in the protocol and the communication and in the optimization stuff, which of course is valuable not just for me, like a researcher right now, but for the future and for trying to endeavor to other students and to be a mentor, I guess.
Ivette, thanks so much. I don't have any more questions. I don't know if you would like to add anything to this conversation that haven't been discussed so far.
No, I mean, it's just, I guess, to say that, again, this protocol, even when it seems to be just for Botryococcus species, it could be optimized and used for another species of microalgae plants. I will say fruits as well. I know some people may work with fruits that have a lot of polysaccharide or like other molecules that they are trying to remove. So if I'm here, I'm open to any comments, suggestion, or questions. If I am, if my expertise or my experience will help someone else, I will be happy to be reached out.
Thank you, Ben.
Thank you so much, Gabriel.